everybody. Today I am going to discuss about the topic gene expression in prokaryotic cells. Gene expression in prokaryotic cell means bacterial cells. Now, what happened? We know that all the enzymes are basically protein, and they are produced. These enzymes are required for our metabolic activity, and for our life. Enzymes are very important, and these enzymes are basically protein, and these enzymes are produced through the same pathway of the protein synthesis, like transcription, translation, like that, and they will be produced. Now question is that the enzyme will not be produced in our body every time. If we do not require, our body do not require enzyme, cell will not produce the enzyme, because there will be wastage of enzyme. So when it will be required, the body will trigger the cell, our body cell, to produce this particular enzyme, and this is called the gene expression, to produce the particular type of enzyme. Like in our room, when we are staying at a room, in a room where we switch on the light. When we go out of the room, we generally put off the light to save the energy. Same thing like that in our body, our cells will produce the enzyme when it will be required. It will not, all the enzymes will not be produced always at every time. Now enzymes are very important things, so I want to talk about some history of enzyme. In 1878, in 1878, the scientist uh, Pune actually first introduced the term enzyme, and the meaning of the enzyme is a Greek word, and the meaning of the enzyme is the in yeast. Actually, we know that first the enzyme was discovered at that time when some yeast, Saccharomyces cerevisiae, will convert the glucose into alcohol, ethanol, and carbon dioxide. So that time gymase enzyme is to will produce the gymase enzyme to carry on the reaction from glucose to ethanol and carbon dioxide. From that time enzyme is built to the, and for that reason this will be named as a enzyme. Enzyme is a biocatalyst. You know that it speed up any reaction, any chemical reaction, and after the reaction they will be unaltered or unchanged. For this reason they can be used repeatedly. So the enzymes are the biocatalyst which will speed up the, accelerate the chemical reaction. And at that time some controversy was there, big controversy between two famous scientists, Louis Pasteur from France and the German chemist Liebig. According to Liebig, the enzymes are chemical substances which are not associated with living cells. But according to Louis Pasteur, enzymes are inseparable from living cells. So they are contradictory to each other. They were contradictory each other, and actually they were not totally wrong. Both were not totally wrong. And in 1897, another scientist Buchner resolved this controversy between these two famous scientists, and what he has done, he demonstrated the cell-free juice from the yeast by filtration, which contain the active enzymes. So this is the history of the enzymes. So enzymes are generally two types. One is constitutive enzymes, and another is inducible enzymes. Constitutive enzymes are those enzymes. Constitutive enzymes are those enzymes which are produced by the body cell all time, always. Like some metabolic pathway, some enzymes will be required in glycolysis and TCA cycle, Krebs cycle. These enzymes are generally produced all the time because the metabolic activity is going on all the time. So this type of enzyme is called constitutive enzymes, and inducible enzymes is the when the enzyme will be required at the particular time, your body cell will produce that enzyme. Not all the always it will the body will produce the enzyme. This is called inducible enzyme. One of the one of the important example of this type of inducible enzyme is beta galactosidase enzyme. I have already told that beta galactosidase enzyme actually hydrolyzes lactose molecule, one lactose molecule to one glucose and galactose molecule for the digestion purpose, and this is done by beta galactosidase enzyme. So this is an inducible enzyme when lactose is act as a substrate as well as inducer. Lactose will when present when we are intaking any lactose rich product like milk or any dairy products, then our body cell will be triggered to produce the particular enzyme beta galactosidase for the digestion of the lactose, for the hydrolysis of the lactose. So this is called the inducible enzymes. Now, how it will be produced, I will discuss about that, and the gene expression will be happened. I will discuss about that. Actually, there are two type of gene control of enzyme synthesis. One is the negative control of enzyme synthesis, and another is the positive control of enzyme synthesis. Positive control of enzyme synthesis. And what happened in the negative control of enzyme synthesis, I will show you. In negative control of enzyme synthesis, like that, of negative control of gene expression or enzyme synthesis, like that, in case of beta galactosidase, this model was developed by Jacob Monod model, because these two scientists discovered and studied this model in of lac operon. This is called lac operon in the particular bacteria Escherichia coli, in short form, which is called E. coli. Escherichia coli is a very predominant bacteria in our intestine, large intestine, colon. That's why it is called E. coli. And in this bacteria, they have studied this lac operon, Jacob Monod model of lac operon of negative control and positive control of gene expression in E. coli bacteria, and they have explained this very well. How they have explained it, I will show you. First of all, negative control, when the negative control will be there, like gene structure will like that. It is repressor gene, it is promoter gene, it is operator gene O, and this is Z, Y, A. And this gene, these three gene, IPO is called regulator gene. Regulator gene. And these three, ZYA, is called structural gene, which is the main gene product, protein. Structural gene. and this is called, this is regulated, this is called the operon, which is called as the lac operon. This is the lac operon from O to A. And what is Z? Z is beta galactosidase enzyme. Y is beta galactosidase permease enzyme, and A is beta galactosidase, thiogalactoside trans acetylase. As you know that the beta galactosidase enzyme is required for the hydrolysis of lactose into glucose and galactose, for the breakdown of lactose required it, and permease enzyme is required for the transport of lactose into the cell, inside the cell. Permease enzyme will required for that, and A for the trans acetylase enzyme, thiogalactosidase trans acetylase enzyme, its function was not well known, but it has been assumed that this will actually detoxifying thiogalactoside. That's why it is called trans acetylase enzyme. But these main enzyme are very important. First, you transport the lactose into cell, and then the lactose will be hydrolyzed by the beta galactosidase enzyme to glucose and galactose molecule. So permease, same thing happened in the diabetics. Insulin act as a permease. Insulin is doing that in our blood, glucose level, glucose will be there, and they will transfer the glucose into the cell for the metabolic activity, for the breakdown of the glucose. If the insulin will be less in our body, then the glucose will be not transported properly into the cell, and they will be not digested properly, and metabolic activity will be not happen, and in our blood glucose will be more. So that's why insulin, same thing will be done by this Y permease enzyme, and these three enzymes are produced together. And now what happened? This regulated genes, this is the repressor gene. I have told this. This is repressor gene. It will produce the repressor protein. It will produce the repressor mRNA, which will produce the repressor protein. Repression protein like that. And this repressor protein, when there is no inducer in the cell or the lactose in the cell, this repressor protein will bind to the operator gene, and they will stop the production of the mRNA transcription of this particular structural gene. They will stop when the repressor protein will bind with the operator gene. And when the inducer will be present, lactose will be present here. Lactose will be bind with this, lactose will be bind with this repressor protein and make the this type of combined molecule, and they will protect, they will resist this repressor protein to bind with the operated gene, and then your gene expression will be there, and mRNA will be transcribed here, and then your gene product will be produced, enzyme will be produced, and lactose will be digested like that. When the inducer, this is inducer, when inducer will bind with this repressor protein, and they will resist, they will protect the this repressor protein to bind with this operator gene, and then the operator gene will be opened, and they will transcribe the mRNA, and then the protein will be produced. This is called the negative control of lac operon. Negative control of lac operon. And what happened in the positive control of lac operon? In the positive control of lac operon, what will be happen like that? Uh, Actually, when lactose will be converted into glucose and galactose, when glucose will be increased in our cell, body cell, then the CMP, cyclic adenosine monophosphate, this is called CAMP, will be reduced. CMP will be reduced and the gene expression will be not there. When glucose, already the lactose will convert it into glucose. But when the lactose will present, glucose is less in quantity, then what happened? Lactose will more in quantity. What happened? This CMP, cyclic adenosine monophosphate, because this cyclic adenosine monophosphate in glycolysis cycle, glucose will be converted into pyruvate to convert the cyclic adenosine monophosphate to ATP, adenosine triphosphate. So there will be no cyclic AMP in presence of glucose. But when the glucose will be less, lactose will be more, then what happened? The CMP will be more in cell, and this CMP will actually this CMP will activate the cap protein. Cap protein means catabolite gene activator protein. Full form of cap protein is catabolite gene activator protein. And this ultimately this cap will activate the promoter, and your gene will start. Your gene will be started to be transcribed like that. So when the CMP will be there in sufficient amount, it will activate the cap, means catabolite gene activator protein, and this catabolite cap will activate the promoter region of the gene, regulator gene, and thus they start the transcription of the this particular gene or gene product or protein enzyme. So in this way the protein will be produced by the, and this is called positive control of enzyme synthesis. It's these same things are, these are happened in the bacterial cell, prokaryotic cell. Same things will be happened in case of virus. I am telling about this virus like that. When virus like lambda virus, one of the lambda virus, it is generally bacteriophage. It is called bacteriophage, lambda virus of E. coli cell. Bacteriophage means those virus will eat the bacteria. Generally, this lambda virus will eat the E. coli virus, kill the E. coli virus. That's why it is called lambda virus. What happened? This lambda virus first entered the bacterial cell like that. When the first, this is the E. coli cell, and this is the lambda virus like that. Lambda virus, when we enter inside the bacterial cell, what happened? It will generally lysis the bacterial cell to kill it, to kill the bacterial cell. That's why this is called phage virus, lysis cycle. But what happened? Some repressor will be produced like the lac operon. Some repressor will be produced from I gene, and they will block the production of the lytic enzyme for the lysis of the bacterial cell. So generally that time this DNA present in the virus will be integrated in the bacterial gene. This condition is called the lysogenic cycle, and they will block the some operons to produce the particular proteolytic enzyme protein to lyse the bacterial cell. But in the one position, what happened? This repressor protein will be blocked by some inducer. Some inducer will be there to block it, and they will produce the lytic enzymes, and they will lysis the bacterial cell like that. on off. In bacterial cell also this type of operon will be happen is happened generally from both lysogenic cycle and for lytic cycle. Lysogenic cycle means when the viral gene will be integrated in the bacterial genome, it is called lysogenic cycle of the bacteria. The repressor will be active at that time of the viral gene. But when it will be lysis cycle, it will produce the lytic proteinase enzyme for the lysis of the bacteria. It will cause lytic cycle. So there are two type of cycle is present in the bacteria. One is lysogenic cycle and one is the lytic cycle. Both like that. This way the gene will be expressed and protein will be produced, enzyme will be produced, and how it will be controlled, how it will be its production will be stopped. I have discussed it. I hope that you have understood it. So thank you.